Now that we have a handle on the steps of meiosis, we need to talk about its purpose. One of the reasons is to cut the number of chromosomes in half so that you can make sperm and egg cells with 23 chromosomes, haploid cells. But the other one is genetic variation. So let's talk about where that genetic variation comes from. Let's imagine we have a cell with only 4 chromosomes total. So it's a diploid cell with 4 chromosomes, which means that there are 2 pairs of chromosomes. 2n equals 4, n equals 2. Divide 2n equals 4 by 2 and you get n equals 2. So diploid 4, haploid 2. Well, when we have metaphase 1 and those homologous chromosomes are lining up, there's 2 different orientations that they could line up in. You could either have over here on the left, you could have both of the father's chromosomes, father's chromosome 1 and father's chromosome 2 on the left, mother's chromosome 1 and mother's chromosome 2 on the right, or in this image a little bit closer to my window here, you could have them flip-flopped. Father's chromosome 1, mother's chromosome 2, mother's chromosome 1, father's chromosome 2. So you have different possible combinations. Now, each of those is equally likely. And even with a cell that only has 4 chromosomes, this means that we actually get 4 different possible combinations of gametes. You could make, let's say we're making sperm. We can make a sperm cell with dad's chromosome 1 and dad's chromosome 2, mom's chromosome 1 and mom's chromosome 2, so 2 combinations there. Mom's chromosome 2 and dad's chromosome 1, that's combination C, or mom's chromosome 1 and dad's chromosome 2, that's combination D. So if you want to figure out mathematically how many different combinations of chromosomes could show up in any given gamete, what you do is you take the number 2 and you raise it to the nth power, to the n, to the number of pairs that you have. In these cells, we have 2 pairs of chromosomes. So 2 raised to the 2nd power is 4. So we end up with 4 possible combinations of gametes. However, humans have 23 pairs of chromosomes. And when you raise 2 to the 23rd power, you find that every single human being could create 8,388,608 different chromosome combinations. That is 8,388,608 different sperm cells that one individual could create, or 8,388,608 different egg cells that a single individual could create, just based on how the chromosomes line up during metaphase 1. Just based on that random assortment of chromosomes. You could have, 3 of mom's chromosomes show up in one gamete and 20 of dad's, or you could have 5 of dad's and 18 of mom's. You have all these different possible combinations. The more chromosomes you have, the more possibilities there are. Does 8,388,608 not satisfy you for the number of chromosome combinations you could have? Well, good news, because in sexual reproduction, which is after all what these gametes are for, you need 2 to tango. If you wanted to know how many different offspring any 2 humans could create, you would have to multiply one parent's 8,388,608 different chromosome combinations for their gametes against the other parent's 8,388,608 different chromosome combinations for their gametes. If you multiply those 2 numbers together, you find there is over 70 trillion different possible chromosome combinations. That is, any 2 human beings that are trying to create an offspring together, there are over 70 trillion different combinations of chromosomes that their offspring could have. This is why you never see identical twins born in separate pregnancy events. Brothers might look very similar to each other, or sisters might look very similar to each other, but you're never going to get identical twins. The odds of getting the exact same combination of chromosomes in 2 different offsprings is astronomically high, 1 in over 70 trillion. But that's not where the variation ends. There's actually more variation than that, because don't forget, crossing over. In crossing over, random amounts of DNA are exchanged between the 2 chromosomes, meaning that every single chromatid is unique unto itself, and every time we do crossing over, you get a different combination of genes on each chromatid. So when you add genetic recombination through crossing over into the mix, the number of different offspring that any given two human beings could have is virtually infinite. You could keep making babies until the universe collapsed in on itself, and you would never see duplicates. You would never see identical twins born in separate pregnancy events. The process of meiosis involves 2 rounds of cell division. These are called meiosis 1 and meiosis 2. Prophase 1, metaphase 1, anaphase 1, and telophase 1, these are the steps of meiosis 1. Prophase 2, metaphase 2, anaphase 2, and telophase 2, that's meiosis 2. The purpose of meiosis 1 is to create haploid cells. That is, we're going to cut the number of chromosomes in these cells in half. Here I'm drawing cells with 2n equals 2 chromosomes, diploid cells with 2 chromosomes total. That means that the haploid cells at the end are going to have 1 chromosome each. In prophase 1, a phenomenon called crossing over occurs, where homologous chromosomes exchange bits of DNA. This means that some of the genes on your father's chromosome gets onto one of your mother's chromatids, and vice versa. This increases genetic diversity. In metaphase 1, we have random assortment of chromosomes, meaning that it's entirely random which daughter cell is going to receive which parental chromosome. Thanks to the genetic recombination of crossing over, each daughter cell at the end of this process will be genetically unique and distinct from the other daughter cells. In anaphase 1, we are separating homologous chromosomes, so we are cutting the total number of chromosomes in half. However, in anaphase 2, we are separating the sister chromatids on each chromosome that remains. So we are going to break the sister chromatids apart from each other and make them into daughter chromosomes. We'll end up with 4 genetically unique haploid cells. In order to observe the genetic uniqueness of each daughter cell at the end, I'm going to label some of the genes which are being carried by the parental chromosomes. On the father's chromosome, which I have colored in green, I'm going to give him genes for tallness and baldness. On the mother's chromosome, I'm going to place genes for shortness and hairiness, so a short, hairy chromosome. Now, during crossing over, each chromatid is going to exchange bits of that DNA, which means that there's going to be new combinations of these genes on each chromatid. At the end, we're going to end up with a parental chromosome that has the tall, bald genes, another one that has short, hairy genes, another one that has tall, hairy genes, and the last one will have short, hairy genes, just as the maternal chromosome would. After meiosis 1 and meiosis 2 have been completed, these sister chromatids, which are no longer genetically identical to each other, are each going to end up in separate daughter cells, meaning that we will have a sperm or an egg cell with the traits for being tall and hairy, another one with short and hairy, another one with short and bald, and the last one that is tall and bald. 4 genetically unique daughter cells at the end, even though we only had 2 chromosomes to start with.